Hi, welcome back from lunch. I am going to first briefly tell you about the perspective I bring to this question. I'm trained as a statistician, and my mostly computational lab thinks about comparative genomics across a long range of timescales and with a lot of different types of modeling, including the seconds that happen during gene regulation, which we've had some good progress on using machine learning, through to looking at the entire tree of life with stochastic process models. We're primarily interested in understanding humans, but we do that using a variety of different systems, in vitro and across different organisms. To understand the human genome, by looking across different species, I want to highlight two key ideas. The first one is that close relatives will tell us what's different in our DNA. But this alone doesn't really answer the question of why we're human. It is helped by also looking at more distantly related species where we can ask what's the same. And indeed, most of our genome is quite similar, even with distantly related species. And so I have focused on the intersection of those two concepts and have developed statistical tools that I'll briefly describe. First of all, to find highly conserved elements. This was joint work with Adam Siepel, who's now at Cold Spring Harbor Lab when he was a student, and I was a postdoc in David Hausler's lab. And if you're familiar with the conservation tracks in the UCSC genome browser, this and the next tool form the basis for those. We then asked what parts of the human genome are different between humans and chimps, but the same in everyone else. So we looked at conservation actually ignoring humans, and then we unmasked ourselves to the human genome. And the answer is that most of the parts that are very similar between a fish and a chimp are also very similar in human. But then we found a small part of our genome where everyone else is the same and human is different, and we called these human accelerated regions, and Chris introduced them earlier this morning. So over several years, we and others have identified close to 3,000 human accelerated regions. The key is that they're evolving much faster than we would expect in humans, given how slow they are in everyone else. This might be due to positive selection. There's been an adaptive pressure on the human lineage since our divergence from the closest out group in this analysis, chimpanzees, but you could also use an archaic hominin. They might also represent loss of constraint. So you can have evolution through deletion and loss, not just through gain. And also neutral processes such as biased gene conversion that mimic natural selection. So we're not trying to necessarily identify what the evolutionary force was, although I'll get back to that a little bit later in the talk, but simply that the odometer was way below the speed limit and then it went faster, maybe through speed limit to positive selection or maybe just to loss of constraint. This approach was applied about 12 years ago now when I first developed it to the entire human genome and the genomes of a handful of other species that were sequenced at that time, and has subsequently been applied to more and more genomes as more species were sequenced. What I like about it is that it's an unbiased approach. We can scan the whole genome and just see what we find. What was exciting, but also really challenging, is what we did find. And that was that almost all of these HARs are outside of genes. In fact, many of them, about 2/3, are quite far even from a gene. They're not even in the introns or the promoters of genes. They're really in these gene deserts of the human genome, quite far away from genes. This was interesting and suggested a potential role in gene regulation for these fast-evolving sequences, the idea that chimps and humans might be made up of similar building blocks, but that we're using those in different ways. But figuring out what the ways we were building were and how they were different was super challenging from the information that we had at that time, and I think remains a very challenging question today of moving from this interesting evolutionary signature to function and an actual understanding of evolution. So the talk today is in part about patience, of waiting a decade until some tools were available to do what I wanted to do more than 10 years ago, and also about realizing that it's always going to be hard to move from sequence to phenotype, and we should also, as we are in this meeting, be thinking about going the other way as well. So I'm very happy to be here where we're trying to synthesize in both directions. We quickly made progress on a few of the HARs by digging deep on them. The fastest evolving sequence, called HAR1, turned out to be an RNA gene that's expressed in the developing brain from about 17 to 19 gestational weeks in humans. It was actually one of the first long non-coding RNAs to be described before we even had a word, link RNAs, and I spent more months than I'd like to say of my postdoc trying to figure out what this sequence did, because we didn't even really have a concept at that time about RNA genes. They were just starting to be understood. But we figured that out, but exactly what the sequence differences are in this gene and what they mean have been challenging. I didn't take that project with me when I left the Hausler lab, but I know several labs have worked on it. And so while we know how the gene, what kind of a gene it is and where it's expressed, it's still not really clear how its huge number of sequence differences in the human lineage could have played a role in evolution. It's in the right time and place to have played a role, but what that is still a mystery. What was more common amongst the other HARs that have been characterized is that they indeed do look like regulatory elements. An example here of one with an interesting human-specific limb expression pattern. So our hypothesis more than 12 years ago was that many of these HARs are non-coding, these sequences that control the expression of genes. And because the genes that they tended to be close to were mostly developmentally expressed genes, we thought that they might be active during the development of embryos. We second thought that the mutations in them might alter their function as regulatory elements, change the degree to which genes were turned up and down. And that was because of how conserved they were in the other species. It seemed like for millions of years it was important not to change that sequence. So if you did change it, perhaps it would change the function of that element. And if it were an enhancer, perhaps it would change the expression of a gene. And then taking that a little further, we hypothesized that these developmental genes nearby were the targets of those HARs, even though they were acting over hundreds of thousands in base pairs in some cases, and that we might see differences if you were to change the human to the chimp sequence in terms of the expression of those genes and perhaps molecular or even cellular or organismal phenotypes. So that's what we were thinking. But we had really nothing to go on. A lot of the data that is available today wasn't available 12 years ago. And so these were just ideas that we had. And I'll share a story that I remember David Hausler, my postdoc advisor, coming in the room and saying something along the lines of, "Eureka, we figured out what makes us human," when he saw my code on the screen. And I believed it too in that moment. And I think a lot of us did, as we had just, the human genome had just been sequenced, within a year of that date when we had that encounter. And I think we also thought that all the answers to disease were going to be easy to read out of the genome too. And so this is, as we all know, proven much more complicated than we thought. It's not just about having a genome sequence or even just about comparing it across species. One thing that's provided some of a leg up on addressing these hypotheses is the ability to not just read out the DNA sequence, but to look at whether it's expressed or not, whether it carries biochemical modifications or not, whether the proteins that bind to it have themselves different modifications on them, what kind of proteins are there. So all of those kind of assays I'm going to call functional genomics. And my lab spent a lot of time, both for understanding human accelerated regions, but also for understanding the effect of disease mutations and non-coding sequences, trying to figure out what all this data tells us about function. And it's been very important for us to do that with some real biological ground truth. So I'm not going to spend much time on this, but just summarize a lot of work to say that we've been trying to predict whether a sequence in the human genome is an enhancer or not. And doing that, in this case, with the ground truth of an assay where you put those sequences into mice, which has pros and cons, of course, but it is in an organism where you can see the spatial and temporal expression that sequence can drive. The downside is, of course, that it's a mouse and that this is an embryonic assay. In fish, you can also look in adults, but again, it's a fish and not a human. But with that kind of a ground truth, we were able to train an algorithm to predict very well on held out data whether a sequence was an enhancer or not, our novel predictions validated in vivo, not just on held out data. And importantly for this talk, the HARs were highly enriched for predictions of being enhancers, especially in the brain. The next question then is whether these sequences that have human-specific changes in them and that we think are enhancers, what gene in this locus would this interesting enhancer be regulating? We would all probably predict gene C because it's sitting in front of it. But as we've learned from assays studying chromatin interactions, figuring out the 3D structure of DNA, frequently this element is actually regulating a gene that's not the closest gene. We've shown that that's true about 90% of the time, that it's not the closest gene. And so we spent a lot of time both working with labs that measure these kind of interactions and again using computational models to see if we can predict what genes predicted enhancers interact with. Again, not the main topic of the talk today, but in addition to making these predictions, I think it's interesting to open the black box of the machine learning algorithms. And in this case, it wasn't so amazing that we were able to predict these interactions. For me, it was really interesting to figure out what is different about this pairing versus this pairing. And what we found was that the proteins that bind on the intervening piece of the genome tell the story more than what's going on at the gene or at the enhancer itself. So for me, that was the most interesting part of that project. The other interesting part was that we could make predictions about HARs, and we found that they cluster in 3D space nearby these developmental genes that we thought that they might be regulating, even though sometimes they weren't the closest gene or weren't particularly close in the linear genome to each other. So all that, machine learning and terabytes of data certainly bolstered our hypotheses about HARs, but this is still just predictive. And so this line of research sort of slowed down a little bit for me. I felt like we really wanted to be able to show cause and effect and that we didn't have the right tools to do that. And so the lab does lots of other things and we've been focusing on those, but I've been excited about a couple of new tools, new technologies that let us get a little bit closer to actually testing specific hypotheses about whether a change in the genome actually has a functional effect or not, especially in particular for these non-coding sequences, where it's been much harder than for genes. So one of the technologies is called a massively parallel reporter assay. It's a molecular experiment that has at its base this same construct, these same vectors that are used to make those mice that stain blue in the tissue where the enhancer is active. But the key here is that we've figured out a way to do this in high throughput so that we can screen not just a handful of HARs, but many of them. The downside is that besides a tail vein assay that dumps this molecular assay into the liver, so if you're interested in liver, it's great. Besides that, we haven't really figured out how to do it in whole animals yet. So it's done in cells in a dish. And that obviously has limitations. But what's amazing is that we can now test thousands of candidate enhancers in parallel. And really importantly, for what I'm going to tell you, we can put the human and the chimp versions, or even evolutionary intermediates that have some of the human-specific changes in them, all into the same experiment. So we don't have any batch effects, and we can look at them side by side in a dish of cells. And the readout is very quantitative. It's via RNA sequencing, where we look at whether a particular molecular barcode that we know is associated with a particular sequence is seen in the RNA-seq or not. So that's one technology. The other one is that because this is an in vitro assay, I'm pleased that the ways to make cells of very interesting types from both human and chimpanzees has gotten better. It's not perfect, it's not great, but we can now make a large variety of different neural cell types. Here I'm showing neural progenitor and glial progenitor cells with their right markers from human and chimp cells. These are achieved by going through a pluripotent state through reprogramming, and so all you need from a chimpanzee is a fibroblast cell line or a lymphoblastoid cell line, and those are readily available from multiple different individuals. So we now have a way to access in a dish some developmental cell types from the brain in humans and chimpanzees. We're also looking at them in the heart, where my colleagues at Gladstone Institutes make these cellular systems that beat just like our hearts do. And then I'm really pleased to be working with an amazing young scientist, Alex Pollard, who came out of Arnold Kriegstein's lab. And he makes these fabulous four-brain organoids. They have some structures that are interesting from a neuroscience perspective. They aren't brains. But what's been really interesting is that you can use them to generate cell types that no one's been able to generate individually in a dish. So we can get, for example, specific types of interneurons that you don't usually see until the very late embryonic stages in a human or the first few weeks postnatally, which is pretty cool that you can make those cell types in a dish. So we're not necessarily studying these as organs per se, but using them to make, dissociating them, using them to make interesting cell types and then dissociating and sorting and studying those cell types separately. Importantly, Alex and then Tom Nowosky, who's another fantastic young scientist, have been ground truthing all of these in vitro systems with primary cells, so human, not chimp, but human whole hemispheres that they have access to from different embryonic stages and early postnatal stages, and we've been genomically characterizing those with single-cell RNA-seq and ATAC-seq to provide some ground truth for what we see in vitro. So using those stem cell-based systems and using this massively parallel reporter assay, we did the following. So for 2 human and 2 chimpanzee biological replicates, we made 3 technical replicates each of sort of an N2 early neural progenitor cell and a more differentiated N3 neural progenitor cell, and put one of these libraries, these massively parallel reporter assay libraries, via lentivirus into each one of these experiments. And so we have, for 714 HARs, the human version and the chimp version, and we have in each experiment 100 technical replicates of each, and then we have 3 technical replicates and 2 biological replicates of each experiment. So importantly, there's a lot of replication here. As a statistician, that makes me happy, and it was really important to make sure we could get good signal out of this kind of an assay. So back to my hypotheses. Are HARs developmental enhancers expressed in the brain or not? Let me give you where we were a year or 2 ago with these low throughput mouse assays. Through a lot of money and a lot of effort, my lab and a few other labs had managed to characterize 92 HARs. Because we were putting a lot of effort and money into it, we didn't take random HARs, maybe we should have, but we took our best predictions that we really thought were developmental enhancers and tested those. And there was a pretty high success rate. About 2/3 of them show activity in this mouse assay, which is known to have a pretty high false negative rate. So it looked like a lot of them were enhancers, but we'd handpicked them. And we were also happy, of course, that our predictions were validating, but again, with this biased set. By taking 714 random HARs, including some we really thought would be neural active enhancers and ones that we thought wouldn't, we were able to get an unbiased data set. And 43% of that totally unbiased data set, not picked to think that it would have neural expression, 43% of those showed activity in these neural progenitor cells. So just one cell type, 43%. So a lot of HARs are, I believe, active enhancer elements during brain development. Our predictions are still pretty well validated. And interestingly, I think it's important that we did this experimental design with human cells and chimp cells in 2 stages, because there could have been really big trans effects. It could have really mattered which species you did the experiment in or exactly what cell type you picked. Amazingly, we found like less than the fingers on my hand differences between the species of origin or the differentiation stage between these neural progenitors. The information is all in cis in the sequences. So do the human and chimp versions show differences? 2/3 of the HARs that are active enhancers show a difference between the human and chimp sequence that we can say is statistically significant with this amount of data that we collected. So 24 experiments covering these 2 cell stages and the different cell species. You can see that consistency, that lack of trans effects here. If something's blue, it's pretty much blue all the way across. The difference between this half of the plot and this half of the plot is that these are the human sequences and these are the chimp sequences. I'm showing all active HARs here, so for the ones in the middle, it's sort of patchy, right? There's no difference, consistent difference between human and chimp, but you can see a large block that are consistently higher with the chimp sequence. This is the signal or the amount of activity, and these are consistently higher with the human sequence. So there's a lot of cis effects, almost no trans effects for these cell types. What we have seen, and I'm not showing here, is that when you look at very different cell types or a different species like mouse, we do see trans effects. So it's just that these two species and these two cell types aren't so different from each other. The cis effects dominate. And also I want to point out that these fold changes that we're seeing very reliably aren't huge. They are quantitative differences that we can reliably measure that I believe are real, but they are not like on-off switches. It's more like what's happened during evolution is like a dimmer switch going up and down. And I'll say more about that in a moment. Briefly, some of these differences that we've seen in the massively parallel assay, we also can validate in animals. So the question I had was then, let's say you have a HAR, in this case, for this 2XHAR238. It's 357 base pairs long. It's an enhancer. There's some differences. It actually has 9 fixed differences between human and chimp. Did you need to change all 9 of those base pairs or not? I've wondered about that ever since I discovered HARs. They have so many differences. Is there sort of a driver mutation like we see in cancer and the other ones were along for the ride, or were they all important? Did they operate independently, or did one happen and another one compensated? One way to get at that would be to look at evolutionary timing. If we had a lot of intervening lineages along this branch from the chimp-human ancestor, we could figure out whether they happened on different parts of the tree. We have done this analysis using diverse human groups, just 3 representatives shown there, and the archaic genomes. And the answer is that almost all of these evolved before the split with Neanderthal and Denisovan, which is expected because this tree isn't drawn to scale. That branch is pretty long. But it's even more than you would expect by chance. So they're particularly not recently evolved. And there aren't a lot of branches going off here from this branch, from here, right? We have this long branch with nothing else at this point to split it up. So this hasn't told us a lot about the sequence of events, because if I go to a given HAR, almost all of its changes will probably be up here. We've also looked at the evolutionary forces that might have created HARs, and most of them look like natural selection, a case where you've gone faster than the neutral rate on the human branch, but some can be explained by loss of constraint and by other neutral processes like biased gene conversion. Okay, so what we did was we took 7 HARs, and we only did 7 because one of these libraries with 244,000 constructs in it, to get this much replication and to do what I'm about to tell you, you can only do 7 instead of doing 700. What we did is we took each HAR, this one I'm showing you because it's easier to see, it only has 3 human-chimp differences in it. Here's the region of the genome, and you can see everybody else has C, except for chicken as a G and human as a T. Everybody has T, human as a G, and then this base has a little bit more variability, but quite a few of our recent relatives have a T and we have a C. So we made the human version with TGC. We made the chimp version with CTT. And then we made all the possible single base pair mutations, all the 2 mutations, so all the combinations that could have occurred. And then we measured their activity to see what happened. This one's interesting not only because it's simple to explain, but these changes here, the T looks like it created a binding site for a transcription factor in the POO3F family. The G looks like it's sort of slightly unpreferred at that position, but maybe doesn't matter too much. And it's not clear what's going on with this base over here, except that it is a genome-wide association study hit for schizophrenia. The POO family genes have a similar expression pattern. So this is the in vivo pattern in the mouse for this enhancer. It's showing an expanded expression pattern in the midbrain and forebrain, and at this isthmus between the midbrain and the hindbrain, with the human versus the chimp sequence, and the protein expression for a member of this POO3 family shows a similar pattern in the tissues, suggesting that that protein's expressed in similar cells to where we see this enhancer activity. So the story sort of holds together. Now, what happens when we dissect this? This is a little hard to see or to think about, but here are the different versions. I'm showing you this one with only 3 changes, because when there's 10, it gets really complicated. Here's the chimp version, CTT, and here's the human version, TGC. And these are all my replicates, just so you can see if it's consistent. On average, the human one has higher activity than the chimp one. And you can see that changing T at the first position from C to T raises the expression, which would be consistent with this activator binding there, except if there's a G at the second position and a T at the third position. So this TGT kind of lowers slightly expression. So there's an interaction between those 2 base pairs. And you can see that the last position, the context that it happens in matters. So you can imagine that these are happening during evolution and maybe either segregating at the same time in the population or later compensating for each other. So when we think about them, it's important to conclude, and we saw this consistently across all 7 HARs, that the bases don't all do one thing, like all elevate expression. They have a net effect, which is usually pretty small, and in many cases, the different positions are one's cranking it up and another one's cranking it down. So what about getting at the real functional information? Do these changes affect gene expression? You might think maybe not, because they're kind of small fold changes, and gene expression is robustly controlled. We've got shadow enhancers. What's going on? So this is another exercise in patience for me and for you. I don't have the results today, but I'm just going to explain our strategy with you. So we're using these organoid systems to make cells from humans and chimps, and we're using the CRISPR genome editing tool to do deletions, activation, and inhibition of these regulatory elements, as well as to try to edit individual base pairs in them and read out various cellular phenotypes. This is not going to tell us about language. This is not going to answer a huge question about what makes us human, but I feel like it's an exciting step in that direction, where we've been stuck for a long time with these genomic patterns and not really able to get anywhere towards function. So I think just getting at molecular function will be exciting. Finally, in my last minute or two, I want to answer another question. Is having these accelerated regions itself a human-specific trait? So are we unique in having them? And if not, if other primates have them too, do we have more? Is that what makes us human? So after discovering HARs, I quickly took the code, and everywhere it said human, I switched it to chimp. And I saw that I found chimp-accelerated regions, but it's taken a while to really carefully compare that result, bolster it up, and to do it across multiple primates. The reason is it's a very complicated statistical test. You're basically comparing over 1,000 different models for evolution to each other, even with just 6 primates. I'm showing you here the case where nothing happens in anywhere else, any other species, and you're just considering the different scenarios in human and chimp. And this is the graph of possible models and how they're nested inside of each other. I won't talk about the model selection procedure, but it was very important to carefully think about how we conclude what's going on. I'll just show you the result. Modulo genome quality, which definitely contributes to this through false positive accelerated regions, we found pretty similar numbers in the different 6 primates. So everybody's got accelerated regions. Humans aren't special. The other species' accelerated regions are also enriched in regulatory parts of the genome. And each species has something that we observed in humans, which is that the HARs and each species' own elements cluster near each other in the genome, even more than we would expect for conserved non-coding elements, which also cluster. These cluster nearby specific genes, and interestingly, they cluster across species. So if we combine all of them from the 6 primates, and then we just look at random draws of a similar number of conserved non-coding sequences that aren't fast evolving in any of the primates, and we look at how the average distance from one to another, this is the distribution, and these lineage-specific accelerated regions are way over here, on average about 30,000 or 25,000 base pairs closer to each other than regular conserved non-coding elements. And so basically, we all have fast-evolving non-coding sequences. They're different elements in each species. They're not the same element, but they cluster nearby the same developmental genes and pathways, as illustrated here with 10M2 and 10M3 to neurin transmembrane proteins that are important for neural development. Each one of these little black lines on the genome is showing an accelerated region from a different primate. So to conclude, our approach to trying to figure out what HARs are and what they do has been very interdisciplinary, from computational modeling to these in vivo studies, and now these massively parallel genomic assays. And they've been enabled by the stem cell-based systems. I want to thank the contributors and the funders, and I'm happy to take your questions.
Thank you, Kathy, very much. For the sake of time, we'll take few questions. If I can ask, for the very last part, when you showed us the enhancer and the very specific changes between chimp and human, is there any effort to try to do some pull out, like really protein binding and, take it into a more proteomics type of experiment to show the binding and stuff like that?
So the obvious hypothesis then would be that PU3 family, one of those proteins, will differentially bind versus the other. For several HARs, not for that one yet, but for several, we've done EMSA assays, which are competing basically to see which sequence binds the protein better. And we've gotten some nice results consistent with our predictions, but it's not very high throughput to do EMSA. I was just at Cold Spring Harbor a couple months ago and saw a talk about sequencing-based high throughput EMSA. If this is working, we're still waiting to see, but if it's working, this will be really exciting to do.
So we are going back to technology. We need more technology, more advanced.
Well, EMSA is a pretty old assay, but just getting a readout from it that's high throughput is pretty cool. Yeah.
Okay, you mentioned the evolutionary change like a dimer. How does it go together with the issue of the missing link in the human evolution tree?
Tell me what you mean by the missing link.
That you have humans and you have semi-human ancestors that were found, and you have monkeys, and you have many kinds of also archaic monkeys, but you don't have any missing link. Like, how did the monkey become a human?
I see. I think what you're referring to is how rapidly we evolved culturally from our ancestors, that there's sort of a prototype human, and then not very long later we were doing complex burials and cave art, like around 50,000 years ago. Is that what you're referring to? Yeah, it's a great question. I don't think HARs are the answer, since almost all of them evolved before that. I think this is a fascinating question, and it was brought up earlier as well. How much does genetics really tell us, or is what makes us human beyond genetics? I think that my take is that the genetics and these changes in our brain, which are probably real, give us the potential to be modern humans. I don't think they make us modern humans, and I think that culture and other things in the environment besides our genetics are what accelerated that most recent. Yeah, I think we were genetically capable of being modern humans before we were. That's my hypothesis.
Okay, so to save the time, let's thank Kathy again.